Transcription is the first phase of gene expression. Transcription involves RNA polymerase reading a DNA template and producing an mRNA molecule as it reads the DNA template. The process of transcription is followed by the process of translation for protein coding genes. While we're going to focus on protein coding genes, I want you to remember that tRNA and rRNA are also transcribed from their own genes, but that those molecules act as RNAs once they've entered the cytoplasm and fold into a particular conformation. mRNAs will also enter the cytoplasm and be translated. RNA polymerase is the enzyme which transcribes RNA. It transcribes only one of the two strands, called the template strand, while the coding strand is left untouched, but has the same sequence as the RNA that's being made. In the case of DNA, there's a T, while in the case of RNA, there's a U. But otherwise, base pairing rules are the standard base pairing rules, with the exception of U pairing with T in the production of RNA, U pairing with A in the production of RNA. The polarity, or the poles or ends of the molecules, are important. New RNA is always made in its 5' to 3' direction, so the growing 3' end is always being synthesized. RNA polymerase reads the template in a 3' to 5' direction, so that the RNA that's being made is anti-parallel to the DNA. The two strands, RNA and DNA, do not remain attached, but notice that they are anti-parallel, and the DNA or the RNA polymerase reads this template beginning at its 3' end. RNA polymerase incorporates new nucleotides as it reads each base pair on the template. Again, the new nucleotides are added to the growing 3' end. New nucleotides have three phosphates, but two are cleaved, so that the terminal phosphate is the only one incorporated into the growing chain. Transcription begins at initiation. Before the actual spot of transcription, there is a stretch of DNA called the promoter. The promoter doesn't actually encode RNA. It's a place for RNA polymerase to bind, recognize the start of the gene, and recognize which strand is the template strand. Every time that RNA polymerase makes a new RNA from a particular gene, it always starts in the same place and always uses the same template strand. At the other end of the gene, there's a stretch called the terminator. This also does not encode RNA. Instead, it's just a place where RNA recognizes that it's time for transcription to end. RNA polymerase recognizes the terminator and also the promoter. Notice that RNA polymerase has begun transcribing RNA here, and it starts after the promoter. The promoter localizes RNA polymerase, but is not actually part of the protein coding region. RNA polymerase begins transcription, RNA in pink, after the promoter. Elongation of transcription follows initiation, and elongation is just RNA polymerase adding free RNA nucleotides one at a time. It unwinds the DNA as it moves toward the 5' end of the template, and it's always adding to the growing 3' end of RNA. This will continue until RNA polymerase reaches the terminator sequence in the gene. The terminator sequence is the sequence that causes RNA polymerase to detach from the gene and release the completed RNA. Neither the terminator DNA is not a stop codon, and the promoter is not a start codon. Remember that start and stop is for translation, not for transcription. And so the start and the stop codons are somewhere within the RNA. These are going to be recognized by the ribosome to begin translation and to end translation. Beginning transcription and ending transcription is completely different, and it's based on the recognition of the promoter and the terminator. Termination is best understood in bacteria. In this case, RNA polymerase has transcribed an mRNA, and part of the mRNA that's been transcribed by RNA polymerase encodes a sequence of RNA nucleotides that will base pair with themselves, like A pairing with U or C pairing with G. This base pairing within the mRNA forms what's called a hairpin loop. The hairpin loop disrupts RNA polymerase, and RNA polymerase dissociates from the DNA and releases the mRNA. Termination is different in eukaryotes. It's a lot more complex, but it is known to be associated with the processing of mRNA in eukaryotes. Eukaryotic mRNA needs to be processed in 3 ways before it leaves the nucleus. First, it's given a 5' cap, and this is a modified nucleotide that is essential for export out of the nucleus and also for initiation of translation. Second, non-coding regions called introns are removed by splicing, and these exons, the coding regions, are spliced together. Introns don't encode amino acids, and so they must be removed. Now they are important, and there's a reason why we have them, but we won't discuss that in this lecture. Ultimately, all the exons will be spliced together, and this stretch will contain all the important codons for making the correct polypeptide. Also before export, the mRNA is given a tail, and this is a long chain of A nucleotides. It's called a poly-A tail. The poly-A tail is also important for initiation of translation. It and the 5' cap also create stability for the mRNA in the cytoplasm. They protect the mRNA from degradation so that the mRNA can be effectively translated. 3 steps: capping, tailing, and splicing are important for eukaryotic mRNA before they exit the cytoplasm in order to be translated. Transcription occurs in the nucleus, and translation occurs in the cytoplasm. Now in bacteria, everything occurs in the cytoplasm because there is no nucleus. In addition, there's no processing of RNA, and as a matter of fact, in prokaryotes, translation can begin while the mRNA is still being transcribed, which is not the case in eukaryotes, of course, because transcription has to be completely finished in the nucleus, mRNA exported, and then translation started. Now it's time to look at the basics of translation and how polypeptides are synthesized using the sequence in mRNA.